is about efficiency. And in my talk, we are talking about the statistical and design issues and how can we make these designs even more efficient. This is the outline of my talk. I'll first start with the operating characteristics, and I'm going to focus on the type 1 error rate because it's of the interest to the funders and reviewers. And one aspect of the efficiency actually coming from the choosing the right operating characteristics. And then introduce MAMS selection design, some recommendations and conclusions. Let's start with the first part. As I mentioned, there are two parts to my talk. The first part is about the operating characteristics, particularly on the type 1 error rate, and the second part is about the MAMS selection design. Like any other trial, we are interested in the probability of false positive, which is the type 1 error rate, and it is the focus of regulators and reviewers. Another aspect of the operating characteristics, which is of interest to the funders, is power, probability of false negative. And of course, the sample size, which is driven by these parameters. And in my talk, I'm mainly focusing on the maximum sample size because I assume interim stopping for lack of benefit for the designs that I'm going to go through, MAMS designs with interim stopping boundaries for lack of benefit, and the primary analysis, efficacy analysis, is done at the final stage. So I'm interested in that sample size. So let's start with the type 1 error rate. In designs with multiple arms, or designs in general that address multiple research questions, there are two main approaches to quantify type 1 error rate. That is the probability of false positive. One is the pairwise error rate, which quantifies the probability of false positive for a specific comparison or a specific research question, regardless of other research questions that you have in your protocol. Another one is the family-wise error rate, which controls the probability of false positive across all the research arms or comparisons and all the stages. And in the confirmatory setting, we usually need to control this at a pre-specified level. If you have a confirmatory trial, you have to control that at a specific level, and that is usually 2.5% one-sided, 5% two-sided. And in all my comparisons that I'm using in this talk, I'm targeting a type 1 error rate of 2.5% one-sided. When I'm controlling the pairwise error rate, I just say, this is the pairwise error rate control. When I'm controlling family-wise error rate, I just say, I'm controlling now the family-wise error rate, but at the same level of 2.5%, one-sided. And the key question, particularly in trials with multiple research questions, and again become more complex when you add a new arm, is which one of these two type 1 error rates I need to basically choose at the design stage in trials that address multiple research questions. And what are the design and analysis implications of choice at the design stage? I think in the last few years, we have reached a consensus that the most important factor in this decision is the relatedness of the research questions that we are trying to address. And how different are the research arms? If they are related, for example, if your interventions are of different doses or duration of the same treatment or intervention, then the focus of the design should be on the family-wise error rate control. If they are different, if they're targeting different, let's say, if they have different mechanisms of action, then the pairwise error rate should be the focus of the design. But what about the implications when we make our choice based on these principles? I'm going to use the Stampede trial at this stage. That is the original Stampede design, which included 6 arms, 5 pairwise comparisons. So we are not in the adding arm stage yet. We are designing Stampede as a 6-arm trial, which includes 5 pairwise comparisons. And at the design stage, when they did the design of the trial, Each of these pairwise comparisons addressed a distinct research questions. As a result of that, each hypothesis was basically powered as a distinct hypothesis, and the control of the pairwise error rate was the focus of the design. If you want to have some comparisons based on the pairwise error rate control within the MAMS framework and compare it with 5 two-arm trials, we can see the sample size. In both of these, we are targeting a pairwise error rate of 2.5%. So the significance level for the primary analysis is 2.5% one-sided. If you see a p-value of less than this, then you claim efficacy. To run Stampede, the original Stampede, as a 5 two-arm trial, separate two-arm trials, you need 2,580 events across all research and control arms to get a power of 83%. That is the effective sample size, because we have the time to event outcome, then that is the effective sample size, that's number of events. Within MAMS, look, to gain the same power and under the same significance level, look at the effective sample size. That is why we use MAMS design. That is why it is efficient. This is plainly obvious. We are targeting the same power pairwise error rate as it was done in Stampede. And you can see the extended efficiency, the effective sample size reduced by about 48%. However, if the target of the, if the focus of the design was the family-wise error rate, which was controlled at the same level, 2.5% across all comparisons and stages, look at the effective sample size now. It is still more effective despite controlling for multiple testing. It is still more efficient than running separate trials, but it is less efficient than targeting a pairwise error rate. And another side effect of that is you need to go and correct for the final stage significance level for multiple testing. Now, your p-value threshold at the final stage for the efficacy analysis would be 0.0055. And I use the Dunnett's correction, which is one of the most efficient multiple testing procedures to get that value. So if you see a p-value of less than that, then you claim efficacy. So we can see that there is a big difference in sample size and efficiency, whether your target type 1 error rate is pairwise or family-wise. So we saw that the family-wise error rate is driven, in fact, by the number of pairwise comparisons. If you look at that table, that significance level is nearly 5 times. If you times that to the number of pairwise comparisons, you get 2.5%. You get a bit of efficiency, you relax that, and you end up with 0.0055 because I used the Dunnett test. So it is driven by the number of comparisons and requires more strict significance level for the primary hypothesis. You need larger sample size, and the gain efficiency will be reduced if you don't need to do it, and you do it, particularly when you are addressing distinct research questions. Now let's move to the MAMS platform trials, where we have a master protocol and multiple research questions are addressed now in this new paradigm over time, and you have flexible features of adding and dropping. So now an example is a Stampede that turned into platform trial later on, and we are now within this context of the flexible paradigm where you can add new arms. Let's have a look at what the how a type 1 error rate can be, in fact, calculated and controlled in that context. Similar principles apply. A major consideration when adding a new research arm is you need to think how the new research arm, how different that is to the ones already you have in your platform or the existing ones. And of course, you need to get scientific and regulatory reviews and approvals. If none of the above considerations justify the control of the family-wise error rate, then the pairwise error rate should be controlled. The implications of the family-wise error rate control when adding arms, that means controlling the false positive, the probability of false positive for both the existing arms and the added arms. You need to think carefully whether you need to do that. And that depends on the research questions that you're posing. And you cannot control, in fact, for a platform, the family-wise error rate for a platform trial when you don't know how many arms you're going to add to that. And there are design implications for the existing arms. As you saw, we need larger sample size. You need to go back and recalculate the critical value or the significance level for the primary test that you're using. They would be more strict, larger sample size for existing comparisons, and extending costs and trial timelines. But the advantage of pairwise error rate is that there's no impact on the existing comparisons. Simplifies the design and analysis of added and ongoing comparisons. New research arms can be added when they are practically become available. So logistically and statistically becomes much, in my opinion, easier. And improved transparency, consistency, comparability. I mean, some of us who are familiar with the multiple testing, multiplicity issues, we know that there's lots of things going on behind the scenes that we are not sometimes transparent. What significance level we choose, what is the critical value. This improves both consistency and comparability. So the recommendations for the first part is that in MAMS platform trials, whenever possible, try to address distinct research questions. As Max said, don't tinker around edges. Don't just go back to the treatment that you had and try to just, sometimes you need to do that, but you have to think carefully. This provides the rationale for strong control of the pairwise error rate. And it is too challenging to control the family-wise error rate in the case of adding arms. You need to limit the number of arms and also pay a high price. And you don't need to pay that if you choose distinct research questions and interventions. But in some situations, we all know that in practice, things are not as simple as we think in theory. Some situations, you need to control the family-wise error rate, which is the next part of my talk, which I now move on to the MAMS selection design. I think we are familiar, as Max was suggesting, with the standard MAMS design. You compare several research arms with a single control, and you have stopping boundaries for lack of benefit. In my case, for example, as I assumed at the beginning, in this case, I only have 2 arms that reach the final stage, and the rest are dropped at different stages at interim analysis. And if you are in this paradigm, depending on how these interventions are performing, you might end up with different, actually, number of arms ending at the final stage for the primary analysis. So there are 2 issues with that. First, the maximum sample size would have a very large variation in it. And in another scenario, like the Rossini 2, Tommy's gonna talk about it, the total sample size, because the sheer number of the pairwise comparisons becomes too large for any funding agency to actually fund it. So what you need to do in those cases, that you can do, one way of doing that is pre-specify. You have lack of benefit or other stopping boundaries that are for dropping arms, or if you have stopping boundaries for overwhelming efficacy, you might have that. But you might also have an additional rule for selection, and that is to select a subset of arms, a maximum number of arms that you would consider or allow to continue to the next stage. This provides a ceiling on the sample size, maximum sample size, simplifies planning, and also shortens the trial timelines. And you need to because we are in regulatory, in confirmatory setting, pre-specification is key. And all the statistical properties that we are familiar, when you pre-specify, become much more easier. So we have to pre-specify this selection rule. For example, for a 6-arm, 3-stage design that you saw in the graphics, the rule might be 5, 3, 2. You start with 5 comparisons, take the third best based on the criteria that you have, which I'm going to explain, to the next stage, and then final stage, you only allow a maximum of 2. Lack of benefit stopping boundaries might actually reduce that number. And the selection can be based on relative performance, for example, based on the estimated treatment effect, or you can include safety data if you want to. But there are considerations when choosing a MAMS selection design, how to select after stage 1, how many to select, and when to select. We have 2 examples. Let's have a look at examples. I will come back to those questions. My first example is in postpartum hemorrhage, which is an event that happens after childbirth. It's an awful condition, situation, and it's a blood loss of 0.5 liter or more after normal delivery, and 1 liter or more after C-section. And it's a very, quite dangerous, particularly in lower and middle income countries. Women losing their lives, particularly in sub-Saharan Africa, and accounts for about 1/4 of maternal deaths in there. So we have a trial called RED, which investigates the clinical efficacy of 3 different devices against the control arm. And the clinical outcome is the composite binary. So we have a 4-arm trial. Let's look at the interventions or the devices. We have a control arm and 3 research arms. Look at research arm 1 and 2. They are basically the same device with a very minor difference in their functioning. So here, because we are making selection, and also we have this related devices, we need to control the family-wise error rate at 2.5%. So let's have a look at some comparisons. If we run this trial as a 3 2-arm trials and control the pairwise error rate, okay, we are not considering this as running in different times, and you are controlling pairwise error for each of these comparisons at 2.5%. To get a power of 80%, you need 4,200 across. That is the maximum sample size for your primary analysis across all of those 3 2-arm trials. For a standard 4-arm MAMS trial, which controls the family-wise error rate, you are counting for multiple testing. Look at the reduction in sample size. Despite actually accounting for multiplicity, still more efficient. However, the final stage significance level need to be corrected to that value, and you claim efficacy if you observe p-value is less than that, compared with if running it as a separate trial. However, to put that number into perspective, to recruit that number, you need to screen about 1 million women live birthed to get that number. You can see that it's just not feasible. We ideally would like to reduce that number. So one way of doing that is to run it as a selection design. We have a selection stage. We start with 3 comparisons, and then select the best performing arm and continue. And at the end of stage 2, we test the efficacy and include all data in the selection stage as well. So let's have a look at some comparisons. If we run this as a MAMS selection design, we control the family-wise error rate. Look at the reduction in sample size now. And the significance level would be 0.015. And that is because you are accounting for that selection. So you claim efficacy if the observed p-value is less than that critical, or p-value critical value. Another example, which is much more complicated, and Tom is going to talk about that, is Rossini 2 trial in surgical wound infection, where we have, instead of 3 comparisons, now 7 comparisons, starting with 7 comparisons, and it has 3 selection stages, 2 selection stages, and it's run in 3 stages. And the maximum sample size in this case reduced by 25%, targeting the same power and significance and type 1 error. And this design also includes the lack of benefit stopping boundaries. So back to the design questions, how to select after stage 1. It turns out actually ranking these treatments based on the estimated treatment effect is very robust. So you can rank these and select the largest effect, the one with the largest effect sizes. You can include in your selection mechanism the safety data. You can also use an intermediate outcome, but there is a certain criteria that you need to go and read in our article. I don't want to go through, but I'm happy to discuss it at the discussion. How many to select depend on the number of total arm or the research arms. And in trials with 2 or 3 research arms, selecting one arm at stage 1 gives you adequate power. When to select, it turns out that when you have a 20% of the overall control arm sample size gives you adequate power and the control of the type 1 error rate. So the overall conclusions, Max has already mentioned this, consider MAMS selection design when there is clear and strict limit on the number of research arms, on the number of individuals that can be recruited, funds available, or the timelines. Whenever possible, do a MAMS platform trial addressing multiple distinct research questions. And if that is not possible, MAMS selection design is one way to gain some efficiency. Thank you very much for your attention.
Again, any fact questions? We're going to have a full panel discussion. I'll allow one over there. Yes, indeed. What, introduce your name and which institution?
Hi, thank you. My name is Babak Oskooei from GSK. Just one question of clarification. So you're presenting two patterns in a way of the MAMS, and then you have the second one, for which you have to sample less patients because you're constraining the number of interventions between progressive and which is clear. Do you have an idea of the trade-off that you gain in terms of sample size versus the constraints that you have specifying the number of interventions between progressive? What's the trade-off? Could you repeat the question a little bit?
Yes, it is about the number of, my understanding was the numbers that can be selected basically to preserve the operating characteristics. That was the number of, that was your question, I think. If you have, as I mentioned in my, I think in my conclusions, if you have 2 or 3 arms, selection of one gives you adequate power. If you have multiple arms, you might increase the number of arms that you select at stage 1, and you might do it in multiple. The selection stage can be, in those scenarios, can be done in multiple, let's say, stages. Then that gives you more power. But you have to do, yes, your simulations to make sure where that sweet spot is. But if you have 3 arms, selecting one gives you.
Lovely. Thank you very much indeed, and thank you very much.